So basically, I work here at I3S, as Julio said, and I work in the vertebrate development and regeneration group and in the aging and the neoplastic group. And in both these areas, it's really important to understand how the genome translates to function and how function is affected and how we can play with the genome to both to study it and to treat disease or different conditions. So this presentation, I will try to bring some concepts about mutations, about gene editing, and really try to simplify. Sometimes CRISPR is surrounded by this mist of mystery and confusion, and we are going to both try to simplify and talk about these basics, but also try to let you know that things are not super simple. You know, when I was discussing this with Jose Bessa, my supervisor, he told me, you know, just go there, talk about the simple mechanisms and tools regarding to gene edit, but by the end of the presentation, make sure that they know that it's not easy to turn green grass into blue grass, you know, that we can just not make some magic trick and completely change our environment and our surroundings. So I want to do both those things to tell you it's simple. Yes, it's really a revolution in the way we are able to play with DNA, but there are still some challenges, and really having the final effect that we want is not super simple. I will just try to go through some. I don't want to bring history here, but just to let you know how these names come from, the names that we are talking about, because I was told that some of people in the audience maybe not so familiar with some biology concepts. So what exactly is a gene and where these things come from? So the idea of a gene and how traits pass from one individual to the other maybe starts with something that came from, for instance, from Charles Darwin when he talked about pangenesis, this idea that two individuals mixed up their traits, like the mother and the father. There's like a soup, and at the end, you have the birth of a new individual. But at the same time, Mendel was working, as you may know, with his peas, and he was really trying to bring a mathematical thinking for inheritance and for biology, and really try to define how different traits are passed from one generation to the other, and how often there are traits that are kind of hidden inside the organism that are not expressed, but they can come up and be shown in the offspring in future generations. The idea of mutations came a little bit later when Mendel's work was rediscovered by Hugo de Vries and other people. And at this point also, the idea of a gene, of this element that codes different traits, was postulated. A little bit later, Willem Johansen coined the term gene by basically simplifying the pangene concept. And it was the first to talk about genotype and phenotype. Genotype is the set of genes that we have. Phenotype is a set of traits that we actually expressed. Because as you may know, we have generally two different copies for the same gene. These are called alleles. And often we in many cases, we only express one of the alleles. So we have only one of the two traits that we could actually be showing. After that, it started a revolution in genomics, because we start to understand with Thomas Morgans and others and its followers that genes are inherited in chromosomes, and they called for mutations. The identification of this mutation that is associated with the white eyes in Drosophila in these flies was one of the first examples. In parallel, people were starting, so remember that at this time, this was just concepts. People were talking about mutations based on, in the case of mutations, it came from different traits in flowers. They realized for the same species, there were some different traits, and people called that mutations without really knowing what was behind. And when people talked about genes, it was about the transmission of information from between parents and their offspring, and what exactly codes for the traits. But what was behind it was not known. But a long time ago, actually, the first to identify this thing that is inside the nucleus of our cells was Friedrich Miescher, and he called it nuclein, just because it was inside the nucleus of the cells. A little bit later, Kossel actually was able to identify and separate the nucleic acids, that is the DNA, from the proteins, and to identify the nuclear bases that compose the DNA. And those are the adenine, the cytosine, the guanine, and the thymidine. And these nucleobases basically are the alphabet of life, the A's, the C's, the G's, and the T's of DNA. In RNA, by the way, instead of thymine, we have uracil, but we may talk about that later. After that, fast forwarding a little bit, there was the discovery of the structure of DNA with Rosalind Franklin and other people. And from that, the revolution started. And people started to understand how our information is coded on the DNA. That information is passed to the RNA through transcription. And then that information is translated to proteins in the ribosomes, okay? So DNA, as you know, or you may know, is composed by nucleotides. That is nothing more than this chemical with three parts: a pentose, that is a sugar, a nutritionose base, and a phosphate group. DNA is nothing more than a chain of nucleotides with this phosphate deoxyribose backbone and nitrogenous bases binding each other from different strains. So we kind of have this weak chemical interaction between different bases in which adenine always pairs with imine and guanine always pairs with cytosine. And they form this kind of anti-parallel double helix. And when I say anti-parallel, it means that one of the strains is in this orientation, 5' to 3', and the other 3' with 5'. This is important later on when we talk about designing the SGs and doing cloning and playing a little bit with molecular biology, because when DNA is synthesized and when we generally read the DNA, we do it from 5' to 3'. These annotations are nothing more than annotations of the carbons of the pentose. If you, let me see if you can see my pointer. If you look here, I don't know if you can see, there is the number of the carbon, 1, 2, 3, 4, and 5. Basically what happens is that as the DNA starts to be synthesized, it starts from this first nucleotide that has the phosphate bound to the carbon 5, and it continues by adding a new phosphate and a nucleotide to the carbon 3. So it synthesizes from 5 to 3'. It's kind of the sense of life. This information is through this protein, the RNA polymerase, converted into RNA. And RNA is the transcript that is basically will be used to produce proteins. In the RNA, it's really important for us to be aware of these concepts of codons. Codons are important because they are the ones that are translated into amino acids. Each codon corresponds to a specific amino acid. For instance, if I look for this scheme here, you can see that what our cells are able to do is to convert, for instance, if I have a codon, and codons are composed by 3 nucleotides of G, a G, and a G, this corresponds to a glycine. If I have a G, an U, and an A, this corresponds to a methionine. Okay, so basically in the ribosomes, the cell has the ability to convert RNA into protein by reading these codons. And any mutation, any change in the sequence of DNA that then is transcribed to the RNA that can affect the way the ribosomes read these codons may affect the proteins. And proteins is what really is important for the cells, because they are both the structural and the functional components of cells. They form the membranes, they form the cytoskeletons of cells. They exert different functions by breaking down nutrients or signaling different pathways in the cell. So any change in the DNA or RNA will affect proteins and therefore will affect the functions of cells. So at this point, we can talk about mutations. So mutations is any change to the sequence of DNA. We can kind of divide this in micro and macro mutations. Basically, there are these macro mutations that are nothing more than changes in chromosomes, like deletions of big chunks of DNA or translocations from one chromosome to the other. But often when we talk about mutations, we are talking about, and definitely when you're talking about mutations in the context of gene editing, we are doing so by talking about these small alterations in the sequence of DNA. So small deletions, small insertions, or small substitutions of nucleotides. This may alter the codons that are then translated into proteins and therefore, like I said, affect proteins. Okay, so these mutations are also behind many diseases and definitely behind many of the traits that we have between individuals. So when we think about editing life and editing the DNA to induce some kind of change, is this type of alterations that we may also do to revert some phenotype that we wish to revert. Of course, we now know the sequence of DNA, and we have for like 20 or more years known the sequence of the human genome, and we are adding the genome of more and more species every year. But that is only possible due through the technologies in DNA sequencing. We don't have time to go through what is DNA sequences, but basically there are nowadays many technologies. People now talk about the 4th generation DNA sequencing, but basically, fortunately, now we have technologies that allow us to take very little amount of tissue and sequenced full chromosomes, sequence to full sequence of our genomes. And therefore, we can compare it to reference genomes and check whether or not we have mutations that may give us susceptibility to disease, or sequence different organisms and realize what is their traits, what are the traits that they code. The results of sequencing, in particular Sanger sequencing, that is one of the first and is the one that we generally use when we are just sequencing not full genomes but little sequences, is this type of chromatogram in which we have peaks that correspond to different colors. This is done in machines, doesn't matter now, but corresponds to the signals generated by the different nucleotides. Okay, when we have mutations like in this example on your right, these peaks will have a different color or even a different intensity. In here, we have, for instance, a negative control, a simple sequence. In the middle, we have, for instance, let's imagine that there is a disease that is caused by a point mutation, a single alteration in one nucleotide. But because we have 2 alleles, so 2 copies for the same gene, you can see that this individual that is called a carrier has 1 blue peak that corresponds to a C and a small green peak that corresponds to an A. So probably this individual has 2 copies, one with a C and other with an A. And this is quite common, okay? But in this case that is called the patient, you can see that there's only 1 green peak. So there is an alteration in this individual only has, it has 2 copies of this alteration. So mutations are really important because it are the reasons why we have all kinds of traits, including disease. There are many, many examples of simple and complex mutations that result in different phenotypes. But I just brought here one example, the example of sickle cell anemia, because it is characterized by a single point mutation. And this is a very interesting example. Sickle cell anemia is not lethal. People can live with that, although it's very deleterious for people that have this disease, namely when they live in high altitude or they do efforts. But other than that, they can live with sickle cell anemia. It is caused by a mutation in which this T is substituted by an A. So when you are translating this to protein, instead of having glutamic acid in this position, you have a valine. And this is enough to affect the hemoglobin protein and therefore affect the ability of red blood cells to carry oxygen. What is interesting is that when you look to the frequency of this allele, so of this mutation, there is some places in Africa in which you have up to 40% of the population carrying this allele. And this is quite unusual. When you look, for instance, for people in South Africa, the frequency is less than 1%. So what people have noticed for many decades now, but actually has been demonstrated functionally more recently, is that this mutation can actually be beneficial. Because even though it induces this disease when it's in homozygoty, so when an individual has both copies with the mutation, actually heterozygous people, so people that are only carriers, only have 1 mutation and therefore do not manifest the disease, actually have an advantage resisting to malaria. Because when the parasite that causes malaria infects red blood cells, and tries to divide inside these cells to spread its infection. Actually, because these cells in carriers, in people with just one mutation, are a little bit more fragile, the parasite disrupts the cell, destroys the cell earlier, and therefore does not allow the parasite to infect more cells and spread. So having this mutation kind of has this double sword effect. In one hand, if you have both alleles mutated, you have the disease, is highly disadvantaged. But individuals that are just carriers have this advantage because they are able to resist to more efficiently to malaria. And this is important when you think about editing the genome and inducing changes. You always need to be careful of the unintended consequences of our editing, because often we may think we know exactly what are how mutations and how the DNA sequence translates to function, but there are many cases in which that's not completely true. Often when people, and in this presentation, I'm not going to talk about the ethics of CRISPR and anything like that, but often when people talk about the implications of gene editing, it's always very important to distinguish between somatic mutations and somatic alterations and germline mutations. Somatic alterations, as you know or may know, are the ones that occur only in adult tissues, like in our muscle or in our skin or whatever, and are not transmitted to the next generations, while germline mutations are the ones that occur in germline, so in our gametes, for instance, and that may be transmitted to the next generations. Therefore, if we want to be sure that we are not introducing a new trait, a new sequence in the population, we may want to target just the somatic cells and not the germlines when we think about doing gene editing in vivo organisms. By the way, if you have any question, go ahead. So, editing the genome. What do we want to edit? We may want to edit those mutations that cause disease because they induce alterations in the protein sequence. But there are several examples in which actually it is very important to be aware of the sequence that are the sequence that is outside the amino acid coding nucleotides. The DNA is composed by many parts. The parts of a gene that code protein are called exons, okay? In the middle of exons, specifically in eukaryotic cells, so animals and plants, but not in bacteria, there are introns, and these introns are regions of the DNA that do not code for protein. They are in the middle of exons, but they do not code for protein. During transcription, there is a processing of these RNA molecules, and the introns are excised, so they are cleaved, they disappear, and we end up with a mature RNA that is converted, that is translated to protein. There are also other parts of the genome that serve to regulate transcription. The most important and basic is the promoter. Promoters are nothing more than regions that do not code protein, but allow the binding of regulatory proteins to the DNA. In particular, RNA polymerase, the one that converts DNA into RNA. There are also other regions like enhancers and silencers. This is the thing that I work, but I will resist and not talk about it. But basically, these regions are really important because they also allow the binding of regulatory proteins that affect transcription. And there is a very interesting example. That is the example of lactase persistence. As you may know, there are in many parts of the world, like in Europe and in some regions of Africa, there is this trait that people have, that is they can drink milk throughout their life. And this is not a trait that makes a lot of sense because we are supposed to stop drinking milk as we get old. But fortunately for us, many of us carry a bunch of small substitutions and mutations that may allow us to keep producing lactase throughout our life, and therefore we can break down lactose and therefore digest milk. But these mutations are not in the DNA that codes lactase. It's not like we have more protein or a more stable protein. Instead, they are in the intron of another gene, this MCM6 gene that, as far as I know, has nothing to do with milk and milk consumption. But this region has been suggested to be an enhancer of the lactase gene. So changes in the DNA sequence of this region will somehow increase the production or the transcription of the lactase gene. Okay, so when we think about performing genome editing and doing alterations in the genome, you need to be aware that different parts of the genome have different functions, and we may introduce many changes by playing around with these different regions. Yes, I think that is a question, although.
Fábio, I was just curious looking at the map of your previous slide where you say that about the lactase.
Yeah.
Yes, there, the lactose persistence. Is that known why? So the reason why we have that other, or the capacity to still drink milk while we are growing up? Well, we should supposedly should not be drinking because we should not be digesting the. Is there any study that already says why?
I don't think there is any conclusive study. What this, for instance, in this image, what I try to represent are these different point mutations that have been identified as being associated with the persistence of lactase. And as you can see here, different populations in different parts of the world have different alterations. What people have suggested, but this is just a suggestion, there's no conclusive correlation as far as I know, is that these small alterations in this region make this enhancer region much more active, and therefore you have more transcription of lactase, of the lactase gene. Why we evolved? Well, this I think it's actually, people generally talk about this as one of the most recent known examples of human evolution. We don't know why, but generally biology works like that. There's no reason for biology to happen, it just happens. And if it's beneficial, it is kept. And there are benefits for our ability to keep consuming milk. So probably people that carry these mutations had advantage, they reproduced more, and therefore these mutations spread in the population.
Yeah, I can add something more, Fábio, to that. That is, well, at least Luisa always use this as an example. The areas in which you have more frequency of these mutations are areas in which there was a domestication of cattle. So the milk association, and it's a kind of co-evolution of milk production and some kind of gene mutation that evolved. The advantage was to have more food. Those that have more food somehow, drinking milk because the milk was available there, passed the mutations to the descendants.
Yeah, that's a very good point, because probably people in Asia also often this mutation may appear, but if they don't have milk to drink, there's nothing that would positively select this mutation to be kept in the population. So yeah, there needs to be something that kind of incentivizes this mutation to be kept in the population.
Yeah, and there is clear the 3 zones of cattle domestication there.
Yeah.
Hey, wait. In Finland, we are very heavily on this lactose. I have an intolerance, and it's so common that when you visit Finland and you go and eat in the restaurants, you can be sure that everything is already made from low lactose, whatever you order. You don't have to even ask separately. So it's very typical here. And I remember many years ago, I heard some sort of research that they think that it's actually the older genes, the ones who cannot use the milk, that they are inherited from somewhere from Ural Mountains. And then people were, with the evolution, forced to learn to drink cow milk when they were coming down to Europe more. And that's been sort of said, but I don't know how true it is.
Yeah, but as you can see, you see there Kenya, Tanzania, Saudi Arabia, Europe, and Ethiopia, Sudan, because there were different areas of domestication of cattle. It was in Europe, Central Europe. There was here in Africa 2 sites of domestication of cattle, and the other one was in the southeastern Asia. And different mutations occurred in different places where people start to have milk available.
Yeah.
So the percentage of people that have those traits are not so high as that, even though some areas you can get 100% of people with those kind of mutations, but most of the places are lower than 50%, 30% of people. So there are always people that are not tolerant to lactose, but the mutations are different. So if you have an African mutation, it's different from the European mutation.
Yeah, that's interesting.
Okay. So I hope this is an interesting example of how messing up with the regulatory regions can actually allow us also to manipulate different traits. And of course, evolution is the best at manipulating the different traits so far. So I just put this slide here to really make it clear that when we talk about gene editing, we are not talking about GMOs, but in a sense, we have been doing genetic modification for thousands of years, because when we try to create a new crop by crossing crossing the individuals that have the traits that we want, let's say bigger seeds or resistance to a given disease, we are trying to accumulate in that population the genes and the alleles that have the traits or that code for the traits that are more interesting to us. Of course, we are all aware of GMOs, but GMOs or genetic modified organisms are not necessarily organisms in which we did a precise editing of the genome, because what generally happens is that we take a gene that, imagine, confers resistance to a given disease, and we may take this gene from any species. We can take it from bacteria, for instance, and put it on a tomato, and now the tomato will be resistant to the same disease that the bacteria was resistant to. This is, of course, very interesting and has a lot of advantages. It works very well, namely on plants and vegetables. But generally, when we are introducing these pieces of DNA, we are doing it in kind of a random way. There are ways to be more precise, but generally it's kind of random. So you just take this piece of DNA, generally a gene and a promoter, and we put it inside the genome of the tomato, and that's it. When we do genome editing, we are generally trying to do very precise modifications. Okay, we are trying to correct the mutation that causes sickle cell anemia. We are trying to change this mutation that makes this bacteria green and not blue. We are trying to make little modifications, either insertions, deletions, or modifications that are somehow beneficial for us. Gene editing has been done for many years now, but there are different techniques. For instance, in mice, people have been using DNA recombination for a long time. I will not be, because we are running out of time already, but basically what people do is they collect embryonic stem cells from mice. They in vitro perform a transfection with a piece of DNA with a gene of interest or some modification that they want to do in the genome that has these homology DNA arms. And this is really important, because these regions, basically, what they do is they allowed for repair proteins, a repair mechanism of these cells, to kind of use this piece of extra DNA that we inserted as a template, and to repair DNA using this as a template. As a result, we are able to precisely insert a piece of DNA, imagine this red piece, in the genome exactly where we wanted, because we use this homology DNA, okay? There is positive selection and negative selection of these cells. It doesn't matter now, but people make sure that these cells were edited in the correct way, in the correct place. Then they put it back in the embryos of mice. You end up having a chimera in which some of the cells were edited, others were not. You grew these animals, and you hope that some of the cells are developed to originate the gametes, and therefore you will have some animals that are mutant for whatever alteration you did. The problem with this technique is that it only really works properly in mice embryonic stem cells. Even in human embryonic stem cells, it doesn't work very well. And definitely in differentiated cells, other types of cells, it doesn't work very well because this technique depends on random DNA breaks that occur during cell division of these cells. And because these cells divide a lot and because they express a lot of the proteins that are important for the DNA repair, this technique is very efficient here, but not in other cells in which this is not common. So gene editing in a more general term has been done with nucleases. Nucleases are proteins that cut the DNA. There are different types of nucleases, but the principle is always the same when you want to do gene editing. You cut the DNA. You cut both strands of DNA. As a result, our cell, when detects a DNA break, will try to repair it. And it can do so using two different methods, a non-homology and joining method and an homology directed repair method. In this first method, the non-homology and joining, the repair is kind of random, and it just tries to, the cell just tries to put together the two strands of DNA. But this is a very error-prone method. And often there are small insertions or deletions or even substitutions during this repair. There's a lot of mistakes that happen here. For us, this can be beneficial because imagine that I want to somehow affect the coding region of a gene. Sometimes it is enough to just cut it and hope that the cell introduces some mistake during the repairing. These mistakes may affect the coding of the DNA, may affect the codons and how the cell is able to read those codons, what we call the framing, may introduce a framing shift, and therefore will lead to gene knockdown. However, when we want to do something more precise, imagine to introduce a new gene or to introduce a small alteration or do a very specifically and highly controlled nucleotide substitution, often we want to do so using this homology-directed repair. That is a little bit similar to what I have just told you with DNA recombination in embryonic stem cells, in the sense that there is this piece of DNA in red that we want to introduce, and there are homology arms that are going to serve to guide this template to the right place and to allow the cell to use these templates or this piece of DNA as a template to repair the cutting. And by doing so, by introducing this new DNA, this foreign DNA in the right way, in the right place. People have been using, for instance, meganucleases for a long time. These are bacterial nucleases that have evolved naturally to recognize specific sequences of DNA. People have been trying to use this for doing gene editing, but the problem is that it's very difficult to modify these proteins because you need to do, people don't know exactly how to control the recognition site of these enzymes. So you need to do random mutagenesis and do screenings to find out, and maybe you find out a meganuclease that bind to your specific sequence of DNA. But this is highly random. It takes a long time, it's very time consuming, and it's not at all a practical way to do it. So people have tried to use other techniques like zinc fingers and talons that have much more advantages compared to meganucleases. But I just want to say that even meganucleases have been used to treat human diseases in vitro, okay? To do genome editing in human cells, for instance. But when people start working with zinc fingers and talons, that was kind of revolutionary in the sense that they allow us to be much more precise and to be much faster, because you don't depend in complete random mutagenesis. What basically these both techniques are based on are little peptides that recognize small sequences of DNA. Zinc fingers generally are designed to recognize 3 nucleotides. And what people do is that they have like collections of zinc fingers and they can fuse them together in a sense that they recognize, imagine 20 nucleotides or 24 nucleotides by putting together the zinc finger that recognizes the first 3, the next 3, the next 3, and so on. Fused to this zinc finger, to this peptide composed by these little individual domains, is this FOC1 cleavage domain. And what this technique is based on is that when you have binding of both zinc fingers, these FOC1 domains will dimerize and will cut DNA. And they generally do so in a window of around 6 nucleotides. Okay, so talons are quite similar in some aspects. They are a little bit easier to work on. People were very excited when people start doing genome edit with talons. In this case, generally, the domains of talons recognize up to 1 or down to 1 nucleotide. So basically, you just need to get your sequence, find out which parts of these transcription effectors recognized an A or a C or a G or something like that, put them together, fuse them to the FOC1 domain, design another one to a nearby sequence, and then hope that when you have dimerization of FOC1, you have cleavage of DNA. These techniques were very, by the way, people have also done this. Instead of using this nuclease, the FOC1 endonuclease, people have also used, for instance, transcriptional activators to control gene expression. In this interesting example, instead of having here a FOC1 domain, they have a transcriptional activator. They they constructed zinc fingers that recognize promoters of a protein that is very important to drive angiogenesis, so formation of new vessels. And what they were able to do was to increase expression of that gene with zinc fingers. But as I was saying, both zinc fingers and talons were being widely used. There are even gene therapies that use zinc specifically zinc fingers, but there was a lot of interest also in talons. But around 2013, there was a revolution. And even though nowadays people still use zinc fingers and they still use talons, the development of CRISPR-Cas9 has been completely revolutionary. And nowadays, that's the main technique that people use to edit the genome. CRISPR has a lot of advantages because instead of having to construct a specific protein that recognizes a specific sequence of DNA, you have a protein that is Cas9 that is always the same, and you just need to combine that protein with a 20 or 18 nucleotide sequence of RNA that will drive this nuclease to cut the DNA. It also depends on interactions between this RNA and the DNA that are much more predictable than the interactions between these proteins and their targets. It's very easy to multiplex, so to target different regions with CRISPR, while, for instance, zinc fingers are known to be toxic when you try to target many regions at the same time. A recent study has shown to have been able to target more than 13,000 regions in a single cell at the same time. And this is also much more feasible because you only depend of one protein and a small piece of RNA, and it's much more affordable. But what exactly is CRISPR? At this point, I think many of you may already be aware of what it is because it's quite famous now. But basically, it was found many decades ago, actually, in bacteria. And people started studying it because it had this very weird sequence, a lot of repetitions and genes that no one was very aware what they worked for. But people have found out that there are several Cas genes. And basically what they do is that when the bacteria is infected via virus, some of these Cas proteins are able to recognize this DNA, recognize specific sequences in the DNA of the virus, cut it, and integrate it into bacterial genome. The next time the bacteria is infected, the bacteria will express some other proteins, namely Cas9, and will express many of these different fragments. And by combining these fragments in RNA with Cas9, they are able to drive Cas9 to the genome of the virus and cut it. And by cutting the genome of the virus, you are basically preventing it from infecting the cell and basically commanding the cell functions. These regions here are in different colors that came from different viruses, are called spacers, and generally they are separated by very repetitive blocks of repetitive sequences. What exactly? So let's now demystify Cas9. Cas9 is nothing more than a protein that cuts DNA. But this protein has a very interesting, is part of a very interesting complex. because it binds to two pieces of RNA, a CRISPR RNA that basically binds to a target region in the genome that may be the viral genome, but may also be the genome of any cell that we are able to put the CRISPR complex in. And also a transactivating CRISPR RNA that basically serves to allow the binding of this RNA to the Cas9. In around 2013, when people start using this in human cells and in other species, people have introduced here a linker. So it's basically these 4 nucleotides, and they have linked the CRISPR RNA to the transactive RNA. So nowadays, when we want to create a specific piece of RNA to drive Cas9 to that location, to that target location, we just need this. We need a spacer that is around 20 or 80 nucleotides, and this single piece of DNA that often is called a scaffold. Together, this is called a single guide RNA because it's a single molecule of RNA. The only thing that we need to care about when we are targeting Cas9 for all the different locations in different species is this spacer, okay? We just need to go to the DNA, take this sequence, place it together with the scaffold, and there we have it. We combine it with the same Cas9 that we can use for all the cells, all the species, and that's it, okay? Pretty simple. There is a catch though. Cas9, even though we can basically target all regions very easily, it depends on the existence of a PAM sequence or a protospacer adjacent motif. This region, for the first Cas9 that was discovered in the species, the Streptococcus pyogenes, and therefore is often called the SP Cas9, is an NGG in which N stands for any nucleotide, can be an A, a T, a G, or a C, and GG stands for 2 guanines. So you can select any target region as long as right next to it you have an N, so N nucleotide, a G, and a G. Okay? This PAM is not part of the spacer, but still it needs to be there because it allows Cas9 to properly bind and open the DNA. Okay, it has to do with the specificity of the Cas9. Fortunately, as you may imagine, there may be some examples in which we don't have an NGG right in the place in which we want to cut or introduce some kind of alteration. So in recent years, there's been, as I said, a revolution and an explosion in the development of the CRISPR-Cas9 system. And people have found Cas proteins and proteins that act like Cas proteins in many species. And they also have done mutagenesis and different alterations in vitro and come up with a bunch of Cas9. And there's really a lot. So here I'm just putting, I know you probably can't read it, it's fine. It's a non-exhaustive list of different Cas proteins. I just want to highlight, for instance, this one. There is the Cas9NG. When this was published in the same paper, they actually published several variants. But the NG Cas9 is basically a Cas9 that instead of recognizing an NGG as a PAM, it recognizes an NG. As you can imagine, that increased a lot the regions that we can target. But in the same article, they actually presented variations and showed that this Cas9 can bind many regions using as PAM and NAC, NTG, NTT, NCG, etc. So basically nowadays, there are Cas9s that can target many, many regions. There is this example that a lot of people also like, that is the CPF1. I think it's also called Cas912A, that basically the PAM is a TTN. And it also was very interesting because it allows to target repetitive regions in the genome that have no Gs. And I'm just lastly highlighting this Cas13. There is a family of Cas proteins that instead of targeting DNA, allow us to target RNA. And that has some advantages that we may discuss later if you want. So nowadays, I will just show you some of the varieties and flavors of Cas9. Please let me know if you have any doubts or if you want to discuss the function and the elements of the CRISPR-Cas9 technology in particular. As we already discussed, Cas9, you just need the Cas9 and the single guide RNA to drive the Cas9 to its location, as long as there is a PAM there. Nowadays, we have many PAMs. When the Cas9 is in the right place, it cuts the DNA, and the repairing of the DNA can be done in two different ways. If you have non-homologous end joining, we may introduce insertions, deletions, or induce framing shifts that can knock down genes. But we can also recur to the homology-directed repair by introducing a template piece of DNA that can allow us to do precise modifications in the genome, such as nucleotide substitutions or even knocking in a specific gene. There are also many varieties that depend on different activities and use Cas9 not necessarily to cut to DNA, but to do other things. For instance, an interesting modification was done in one of these Cas9s that is called a nicase. So instead of this Cas9, instead of cutting the DNA in both strands, cuts in only one. And this is the advantage of not inducing a double-strand break. So you don't really cut the DNA. But what you can do is to use two different nicases to about the same place. And by using these two nicases, you basically cut DNA in both strands and you can open. Why would you want to use this? Basically, because of off-targets. And that is the main problem when you use CRISPR-Cas9, is that even though people now have come up with highly efficient, highly specific Cas9s, there's always the risk that when you select that target, those 20 nucleotides, and you are trying to drive Cas9 to that location, you may actually be cutting and inducing mutations in other regions of the DNA. Nowadays, every time when you design SGs, RNAs to target a specific region, generally the online tools in which you design these SGs, they tell you what are the potential of targets, and you need to be aware of those. And often you should actually check if by trying to introduce mutations in your target, you are also affecting these off targets. By using nicases, because you depend on two Cas9s, you reduce substantially the risk of inducing mutations in other regions, because you need both cuts in these both places. And it's highly unlikely that you would find another region in the genome in which you would have these two target regions very close by. People have also come up with other things using dead Cas9. A dead Cas9 in which is a version of Cas9 that does not cut the DNA, not even once, not even one string. So why would you want to use a Cas9 that does not cut the DNA? Well, because it allows you to drive different things to specific regions of the DNA. Some of the most interesting ones are base editors. Basically, what this is a Cas9, a dead Cas9 that does not cut DNA, that is fused to different base editors. That basically what they do is to induce substitutions. They can induce substitutions of an A to a G, or sorry, an A to a G to a T, or an A to a C. Okay, there is no cut here, and basically what this allows you to do is to do substitutions only, and therefore you are sure that you are not introducing small insertions or small deletions or having any framing shift that may compromise expression of genes. People have also fused different peptides to the Cas9, to the dead Cas9, and kind of after fixation. So let me restart. People have fused these tags to the dead Cas9. By using the guide RNA as usual, they are able to drive this Cas9 to specific locations in the genome. They use chemicals to do cross-linking, basically to make sure everything is put together. They are linked, chemically linked. You can then pull down this tag with imagine specific techniques like specific antibodies and bring together all the proteins, DNA, and RNA that is nearby. And it's a way for you to read the DNA and understand which type of regulatory proteins bind to that region, which type of RNAs are being transcribed, etc. People have also used dead Cas9 to visualize different regions of the genome. Imagine that you want to see if a specific enhancer is interacting with a specific promoter. You can tag this enhancer with one color and the promoter with another and see it. And this is basically done by modifications in the guide RNA that form these hairpins, these structures that are recognized by RNA binding proteins that are fused to fluorescent proteins. Okay, so therefore you can kind of combine these different fluorescent proteins having different hairpins, and you can have a full set of colors and kind of have and localize different regions of the genome and see how they are localized in different conditions in the cell. You can also, we are over time, but we can also fuse this dead Cas9 to proteins that modify transcription, either activating transcription or repressing transcription. And therefore, you don't need to cut anything. You don't need to insert anything. If you just want to increase the expression of a given gene, you can use this dead Cas9 fused to an activator of transcription. And there are even versions of these activators that respond to both light and chemicals. So you can make sure that you are only inducing activation of a given gene with this. But coming back to gene editing, more recently, people have been very exciting about a new version of gene editing with Cas9 that is prime editing. In this version, and this is quite recent, as a couple of years, in this version, you cut the DNA in just one strain. But the trick here is that fused to the Cas9 is a retrotranscriptase. And this enzyme basically converts RNA into DNA. And the trick here is to include in this new RNA that is called prime editing guide RNA, the template with the mutation that you want to insert in the genome. When you cut the DNA, the retrotranscriptase will basically convert this piece of RNA into DNA, and through a kind of random repair mechanisms, this mutation may end up being incorporated into the genome. And this has the advantage of being highly precise, does not include exactly cutting in the DNA. And because the template is right there, it is quite efficient. I was going to show you just some examples of things that I have done in my work. Like in this case, I used CRISPR-Cas9 to cut an enhancer region of a gene that is important for senescence in human cells. And I was able to see that I was able to increase senescence in vitro. We also use CRISPR-Cas9, I'm going very fast now, sorry, in zebrafish, for instance, to induce mutation. In this case, I was targeting the GFP protein. I had this line that expressed GFP in the central nervous system. And basically what I did was, just as a test, I targeted the coding region of this green fluorescent protein. And I saw, and of course, this is somatic mutations, that some of the cells lost GFP expression. And this is just some recent publication in which we actually showed that expression of Cas9 in muscle alone is deleterious. So it leads to muscle death. So you need to be very careful when you think about delivering Cas9 and ESG, what may be the side effects, because at the end of the day, we are still introducing bacterial nuclease into our cells, and unexpected effects may occur. I just want to finish maybe here to show you some of the interesting developments that have been done, and show you that good and bad things are happening. In one hand, CRISPR is already being used to treat disease, both by editing human cells with CRISPR in vitro and putting it back into the patients, or by directly injecting the CRISPR components into our tissues. For instance, in this recent case, people were injecting the CRISPR components directly into someone with blindness, hoping to treat this condition. People have also been using in-body gene editing with zinc fingers recently, but you may be aware of, for instance, some of the most bad news about use of CRISPR, in which they actually target the germline, because what they did, these Chinese scientists, was to basically edit the genome of embryos, and that actually basically they were trying to make them resistance to HIV. I think in a way they didn't even succeed on that. and it's quite troubling because more recently it has been shown that trying to use CRISPR-Cas9 to edit human genomes can induce a lot of chromosomal alterations with unexpected consequences. CRISPR has a lot of hope. As you may know, last year, the Nobel Prize of Chemistry was attributed to two of the scientists that contributed to the development of CRISPR and its application. And nowadays there are a lot of startups and companies that promise to use CRISPR to everything, from diagnosis to improving organ transplantation, to, of course, personalized treatments and single treatment cures, to create new crops that will solve world hunger, that will integrate biology into modern electronics, and that will, of course, fight cancer and all of that. So there is a lot of hope. The technology is pretty simple because it's based on just one protein and a small piece of DNA. I had some here examples that I will skip because of time, but there is still some challenges. In one hand, the PAM. As I showed you, there are now many different variants of Cas proteins that allow us to target different regions of the genome and be a little bit more permissive in terms of the PAM. The thing is, if now you have 20 Cas proteins, all depending on different PAMs, then the system is not so simple. So ideally, and that has been, people have tried to been working on that, we would have a Cas9 that would be kind of independent from the PAM. So you would be able to really, with one protein, target any region of the genome. Of course, the main problems, and especially when you think about using this to target humans, it's always being sure that it is highly efficient, that if you target one region to cut it, it will actually cut it, but also that you are not inducing cuts in other places, so to decrease off targets. And the decrease of off targets can be done with nicases, with prime editing, or even some people have suggested that if you are just trying to knock down some gene instead of targeting the DNA, you may target the RNA. And in that case, you are not introducing any permanent change in the genome of the individuals. You are just messing up with transcription. And one of the big challenges is still how to deliver these CRISPR components to an organism. Because in cells, there are many ways. We can do electroshocks to cells and force the DNA to get into the cells. We can use liposomes, so these structures made out of lipids that easily get into the cell. So we can put the Cas9 protein there and the SG is there, and we can make them get into the cells. In, for instance, some animals like zebrafish, we can directly inject the Cas9 protein and the SG in the embryo cell. But if you have a full organism, a person, and you want to treat a disease, you need to come up probably with a virus that are able to infect the cells that you want to target. And that's quite difficult because even though viruses are good at treating and infecting cells, often they don't infect all the cells. And if you have one disease that is mainly caused by a mutation of a gene that is expressed in the liver, you need to put the Cas9 components in the vector that will infect the liver cells. And of course, there is ethical and a lot of social issues. When I think about social, is even if this type of technology will arrive to everyone at the same time, and maybe we'll have some people that will be able to treat diseases and all of that, while others will, because so far this is not super cheap, and others won't. So I hope I have shown you that, yes, this technique depends, and genome editing nowadays with CRISPR-Cas9 depends on quite simple tools, but actually editing the genome and the cells that we want when we want is still not super easy, and there's still a way to go. And there, of course, there are implications, as you may know, with this type of alterations. You may want to make the grass blue, but I don't know if that's necessarily a good idea just because we can.